Hello and welcome. Today I'll be talking about using eDNA as a method of detection of aquatic species. So just a quick recap, what is DNA? DNA is found in all living things inside the nucleus and also inside the mitochondria. So for eDNA studies, we actually use mitochondrial DNA. The reason behind it is because there are multiple copies of that DNA in a cell. It is also more stable in the environment, and it is also much shorter, so it's easier to compare it to already known sequences. Now, what is eDNA and where it comes from? So eDNA actually stands short for environmental DNA, and it comes from anything that's shed by an organism. So skin, hair, excretions, bodily fluids, gametes, sperm, anything that's separate from the actual organism. But just a quick history, so it's quite new area of studying. It only started in the 1990s. Now I know for some of you it's a lifetime. But since then, it has grown rapidly to that extent that in April 2017, there was already 158 papers published with the use of eDNA. But how do we actually do this? So we collect our water samples, then the water is filtered or centrifuged, then the DNA is extracted and amplified and detected by light. Now to quantify our DNA, we actually use what's called quantitative polymerase chain reaction. I know, a mouthful, but it's actually a chemical reaction. So we put our DNA against a primer and a probe, and it works like a big jigsaw puzzle. Basically, if everything fits, then the DNA is amplified, and then we can compare that sequence to sequences from online resources like the gene bank. But what can you use eDNA studies for? So it can be used for detecting rare and elusive species, migration and spawning behaviors, endangered species surveying, management plan actions against invasive species, distribution and movement in water, abundance, and many more. But what are the pros and cons of using eDNA studies? Well, it degrades really quickly. It only lasts 7 to 21 days in the environment. So it's a bad thing, but it's a really good thing if you're looking for something that you don't think is even there. Because if you do get a detection of that DNA, it means that it's a recent sample. Now, the big thing with eDNA for me is that it is non-invasive surveying methods. You do not need to capture the organism that you're looking for. That's a big, big plus for me. Thank you for watching and listening to my introduction to eDNA. This was a very quick run through, but please tune in next week where we will be using eDNA for detecting Arctic char in Irish lakes. Bye bye.